to 66, so overall very well shown there. Okay, so today we're going to cover chapter 17, which deals with the Krebs cycle. The cycle's so nice, they named it thrice. Krebs cycle, TCA for tricarboxylic acid cycle, because the first intermediate citrate is a tricarboxylic acid. It's got 3 carboxyl groups on it. Or the citric acid cycle as the first intermediate, also citric acid or citrate, the iodized form. Right? Krebs for the person who discovered it. So just got a bunch of names. That's how important this cycle is. Okay, this cycle both is responsible for fully oxidizing the remaining carbons that we have in our fuels from glucose, and all of the carbons actually that we get from fatty acids, right, where they're fully oxidized down to carbon dioxide. And it's this oxidation is going to power the reduction of NAD+ to form NADH, which we'll see later on in chapter 18, is going to provide the energy to actually phosphorylate ADP to form ATP in mitochondria. So just a review, we covered this when we discussed glycolysis, right? So we have our glucose molecule, which we then take all the way down to 2 pyruvate molecules during glycolysis. Through all of this, we get 2 ATP molecules, not very much, and we get 2 NADH molecules as well. Now, the only thing that we've talked about going on with these NADH molecules is what? Where do these NADH molecules go? We talked about only one pathway that's really involved. Fermentation in general, right? Lactic acid fermentation, ethanol fermentation, all other types of fermentations. The only thing that we've done with these NADH. Now we're going to see more NADH coming from the citric acid cycle. And again, when we get to chapter 2, we're going to see how these NADH molecules are used, if not for fermentation for glycolysis, or the citric acid cycle is really only one thing that they can do. Now these 2 pyruvates, again, we discussed what happens to these. Right? Either goes into fermentation to form either ethanol or lactic acid or some other end product. And if there is sufficient oxygen present for reasons that we'll see, right, these 2 pyruvate molecules can also be oxidized to form 2-acetyl-CoA. And I've written down here the number of carbons that we're dealing with. Right? As we go through these pathways, one of the most important things you have to keep in mind is to always keep track of your carbons. Carbons don't just disappear. Right? So we started with 6 carbons there in glucose. We have our 6 carbons still left here in our 2 pyruvate molecules. 2 pyruvate molecules give us 2-acetyl-CoA, each of which, you can see on the board, has 2 carbons. So now we have 4 carbons here. Where the other carbons go, remember, in this pyruvate dehydrogenase complex, we also lose 1 CO2 per pyruvate, so a total of 2 CO2s, right? So we haven't lost any carbons, and now this acetyl coenzyme A is ready to enter the citric acid cycle. And what and where does the citric acid cycle take place? Well, the citric acid cycle takes place in the mitochondrial matrix, here in the yellow, right? So we all remember, right, from Gen Bio, mitochondria have 2 membranes, an outer membrane and an inner membrane. In between, there's the intermembrane space. Now, the outer mitochondrial membrane is permeable to most things, right, except notably protons. That'll be important later. Okay, but the inner mitochondrial membrane is not, right? The inner mitochondrial membrane is impermeable to almost everything. So what we see is when we talk about the intermembrane space, very often what you're going to see it referred to is as the cytosolic side, because the composition of the intermembrane space very closely mirrors the composition of the cytosol, okay, with some notable exceptions that we'll get to later on this semester, right? So when you see in the book or in the literature, cytosolic side, in the context of the mitochondria, really what they're talking about is the intermembrane space. Now the matrix is everything inside of the inner mitochondrial membrane. This is where the citric acid cycle takes place. This is where the electron transport chain is, sort of, right? So here, this is where all of the energy that we're going to get, or most of the energy anyway, from our fuels is going to come from, right? And the inner mitochondrial membrane has all of these folds, right, which you can see here in this electron micrograph here, all these folds, which are called cristae. And these cristae are where much of the oxidative phosphorylation is going to take place, right, as you see. But right now, let's concentrate on the matrix. Since inside is where the citric acid cycle takes place. The citric acid cycle is just a series of redox reactions. There's really nothing special about the citric acid cycle, right, in terms of the types of reactions that are occurring. What is important in the citric acid cycle are the intermediates, because the intermediates of the citric acid cycle can serve as the precursors for many biomolecules, such as heme, such as different amino acids, and so on, right? And it also is going to generate high energy electrons, or what we call high energy electrons. So let's see everything that we get out of the citric acid cycle. Let's going to result in the oxidation of an acetyl unit from acetyl-CoA into 2 molecules of carbon dioxide. So both of these carbons, through actually 2 turns of the citric acid cycle, will be oxidized all the way down to carbon dioxide. It's the final pathway for fuel oxidation, right? So notice, all we've done up until this point is generate this acetyl-CoA and 2 ATP, so really not very much energy. This is the final pathway that all fuel molecules are going to enter, and this is where most of the energy is going to come from, at least in terms of ATP. The oxidation of these carbons generates high energy electrons, which are going to be used to power ATP synthesis, or the phosphorylation of ADP. And this is kind of confusing when we say high energy electrons. There is no such thing as a high energy electron. An electron is an electron. What we refer to as high energy electrons means that the compound on which those electrons exist will release a lot of energy when those compounds, or when those electrons are released. In other words, when that compound is oxidized, it releases a lot of energy. These electrons are going to be carried in this case on NADH and FADH2, the oxidation of which releases a lot of energy. And it's the release of that energy that we're going to capture later on in order to power the synthesis of ATP, which of course is going to require positive 30.5 kilojoules per mole to synthesize. This citric acid cycle also produces precursors for biosynthetic reactions. This is some examples of those. Second, of course, it occurs in the mitochondrial matrix. Right? So here we have an overview of the citric acid cycle. Right? The citric acid cycle removes electrons from the acetyl-CoA. To reduce NAD plus to NADH, we're going to get 3 NADH molecules. Reduce 1 FAD to FADH2. Okay? 2 carbons enter the cycle as acetyl coenzyme A, and through 1 turn, 2 carbons leave the cycle as carbon dioxide, here and here. Right? So 2 carbons in as acetyl-CoA, 2 carbons out as carbon dioxide. Now, as we'll see, it's not the exact 2 carbons that enter, that leave as carbon dioxide in 1 turn of the citric acid cycle. Right? But stoichiometrically, it doesn't matter. 2 carbons in, 2 carbons out. Okay? 2 carbons leave as carbon dioxide. The electrons on NADH and FADH2 are high energy again, due to the fact that the oxidation of NADH and FADH2 is going to release a lot of energy, right? And that there's nothing special about these electrons necessarily, right? So out, again, what do we get? 3 NADH molecules, 1 FADH2 molecule, and 1 molecule of ATP or GTP directly. Now, each molecule of NADH, the oxidation of that NADH back to NAD+, is going to provide enough energy to power the synthesis of 2.5 ATP molecules on average. For FADH2, that's going to give us 1.5 ATP molecules. So an NADH is the same as saying 2.5 ATP molecules, and FADH2 is the same as saying 1.5 molecules. The reason for this, okay, is thermodynamically, the oxidation of FADH2, do you think is going to release more or less energy than NADH? Somebody said it. Less, right? It's going to release less energy, so we can make less ATP, right? So that makes sense thermodynamically. The mechanism of how NADH is actually generating more electrons, again, we'll see in chapter 18. Now, the electron transport chain also exists in the mitochondria, specifically in the inner mitochondrial membrane, right? So the electron transport chain is embedded in the inner mitochondrial membrane. And we see that schematically here. So the electron transport chain is a series of proteins in the inner mitochondrial membrane. Electrons flow from either NADH or FADH2 into the electron transport chain. And of course, we're getting these NADH and FADH2 molecules from the citric acid cycle. These electrons flow into the electron transport chain. The final electron acceptor in the electron transport chain is going to be oxygen. So the electrons flow from NADH to oxygen, reduces the oxygen to water, and oxidizes the NADH and FADH2 back to NAD plus and FAD, which can then go back into the citric acid cycle and accept more electrons. Okay. Now, when these electrons flow through the electron transport chain, that energy isn't released as heat, right? That energy is actually captured by the electron transport chain and is used to move protons from the mitochondrial matrix into the intramembrane space, right? So the pH of the intramembrane space is it going to be higher or lower than the matrix? Lower. In fact, it is about 1.4 units lower in the intramembrane space than it is in the matrix. And what then do we have if we talk about the protons across this gradient, right, what do we have stored then? What is stored in a concentration gradient? Energy, exactly, right? So we have energy now stored in this proton gradient, which we also call the proton motive force, sometimes referred to as delta P, right? And so we can release this energy. It's about 21, 22 kilojoules per mole of energy stored in this proton gradient. We can release this energy either by just going across the membrane, right? So poking a hole in the membrane. What's going to happen if we do that? Well, the protons flow from the intramembrane space into the matrix. And where is all that energy going to go? It's going to be released, obviously, as... How is energy usually released if we don't capture it? Heat, right? So we don't want to do that, right? We don't ever want to just release energy usually as heat. Instead, we want to capture it. In this case, we're going to capture this chemical potential energy by only providing a path for these protons to re-enter the matrix through ATP synthase, right? Here's an enzyme ATP synthase. The protons pass through here, releasing energy into the enzyme ATP synthase, which then uses that to phosphorylate ADP to form ATP, right? So it is the production of these NADH and FADH2 molecules, right, that are carrying energy to the electron transport chain, which then store energy in a proton gradient, which is then released through ATP synthase, which is what powers the synthesis of ATP. And where did we get those NADH and FADH2 molecules from? Well, we got them from the oxidation of acetyl-CoA. And where did we get this acetyl-CoA from? We got it from glucose, as we've seen. We'll get it from fatty acids. And we also got it from amino acids. So it's the oxidation of fuel that provides enough energy to make these electron carriers, NADH and FADH2, which then releases the energy of those electrons through the electron transport chain and generates this proton motive force, which then is able to power the synthesis of ATP, right? So let's begin the citric acid cycle. Right? So we're entering the citric acid cycle with acetyl-CoA, we see that here in red, and oxaloacetate. Again, the only place that we've seen so far that we can get acetyl-CoA is from pyruvate dehydrogenase. Later on, we'll see more sources of acetyl-CoA, right? And this is going to condense with oxaloacetate. I'm going to draw it the way I draw it because it makes it a little bit easier to follow. Acetyl coenzyme in condenses with oxaloacetate to form citrate. Right? So we have 2 carbons, 2 carboxylates. Now we just have a 3rd carboxylase. Citrate is catalyzed by citrate synthase, right? Now for this we also need some water, H2O, right? And out, what should we get? Well, all right, we've got the 2 carbons from acetyl-CoA right here, right? Water right here, but what else do we need? What else is coming out from here? What should be coming out? We haven't accounted for one thing. Right, coenzyme A. So we just release coenzyme A, write it like that, coash, because there's now a free self-hydral at the end. That's exactly what we see here, right? Oxaloacetate plus acetyl-CoA goes through the citryl-CoA intermediate. Notice we have a thioester here. This thioester is still high in energy. We can hydrolyze this, right? So we can hydrolyze this off, releasing the coenzyme A, and now we have our tricarboxylic acid citrate, right? This is the first intermediate in the citric acid cycle. Again, this proceeds through an aldol addition plus a hydrolysis. So aldol addition we've seen reverse in glycolysis and a normal aldol addition in gluconeogenesis. This is exactly what's going on here. And then a simple hydrolysis which releases the coenzyme A, giving us our citrate. Okay? Now, citrate synthase, right? We see here, and it always exists as a dimer. And it's going to do something that we've seen another enzyme do before, hexokinase. All right, so here, citrate synthase exists as a dimer. It binds oxaloacetate first, right? When oxaloacetate binds, we see the conformation change, right? So you see we go from the left to the right. It's a significant conformational change. And when we get this conformational change, now we form the binding sites for acetyl-CoA, right? So acetyl-CoA cannot bind to citrate synthase until oxaloacetate binds. And why might this be important? Well, I already gave you a hint, right? This is doing a similar thing that hexokinase did. Remember, hexokinase also changes its conformation. And why did hexokinase change its conformation? It's trying to exclude something from the active site.
Like allosteric regulation?
No, not allosteric regulation. It's trying to exclude something from the active site. Something that's very abundant and something that we don't want. Water, right. Because what would happen if we had water in the active site? What type of reaction would occur? Hydrolysis, right? Well, if we come over here, if acetyl-CoA binds and there's water in the active site and we get a hydrolysis reaction, what are our products going to be? Well, what's going to be hydrolyzed? Well, we have a thioester, which is unstable. So chances are that's what's going to be hydrolyzed. So what would our products be if we just allowed acetyl-CoA to be hydrolyzed? Right, coenzyme A and acetate, the ionized form of acetic acid, right? Which is not useful, right? We can't really do anything energy-wise with the acetate. So we have to bind oxaloacetate first, right? And then form the binding site for acetyl coenzyme A. This means that every time an acetyl coenzyme A binds a citrate synthase, there's already an oxaloacetate in place to go through the proper mechanism, as opposed to allowing water to enter the active site and catalyze the hydrolysis of acetyl coenzyme A, resulting in wasteful, byproducts of acetate and coenzyme A. Right? So if we look at the mechanism for citrate synthase, it happens in 4 steps. Step 1, histidine, in this case, position 274, donates a proton to the acetyl coenzyme A, and this promotes the removal of the methyl proton by aspartate 351 over here. 375, sorry, my numbers mixed up, right? So here, histidine 274 donates a proton, right, which promotes the removal of one of these methyl protons by aspartate 371, which again is kind of weird, because how do we expect aspartate 375 to behave? What do we know about aspartate residues? They end in a carboxyl, so how do we expect them to behave with regard to protons anyway? Yeah, they donate. Why? Yeah, they're acidic. They got a pKa of about 2 or 3. So we wouldn't expect it to be accepting a proton here. So we've seen a similar thing happen before, right? Where have we seen a similar thing happen? Aspartate or glutamate acting as a base. Yeah, right, so we've seen this mechanism before. All right, so now oxaloacetate is activated by the transfer of proton from histidine 320 now, right, to its carbonyl carbon atom. So here we've dealt with the acetyl-CoA, we've activated it. Now we activate the oxaloacetate, and now the acetyl coenzyme A can attack the oxaloacetate, right, and form this citryl-CoA intermediate. This citryl-CoA intermediate can then be hydrolyzed to form citrate and coenzyme A, right? And this wouldn't be possible if water were in the active site, right? And it's very likely if citrate synthase could bind either water or oxaloacetate and catalyze the same, reaction. Well, there's way more water than there is oxaloacetate, so we get far more acetate being produced than citrate, right? So here we have our hydrolysis, again, leaving us with citrate, which you see down here, and coenzyme A, CoAsh, right? And we're ready to now enter the citric acid cycle process. Citrate is then isomerized to isocitrate, okay? By this enzyme here, aconitase. Here comes the aconitase. 3 carbons, 3 carboxyl groups. Moving this OH, right? We're just moving the OH up by 1, essentially. And that's called isocitrate, catalyzed by aconitase. Actually proceeds through this intermediate here, aconitase, right? This is where it got the name from. And all this does, aconitase, is it removes the water from citrate on carbon number 3. This gives us, again, aconitase. And all that citrate is going, or all that aconitase is going to allow to happen is for water to be added back. Now, it could be added back to carbon number 3, in which case we get citrate, or it can be added to carbon number 2, in which case we get isocitrate. And all these are reversible. There is nothing in a conatase that's going to dictate where the water molecule goes. So then what is going to determine which way this goes? Are we going to be hydrating carbon number 3 to form citrate, or are we going to make isocitrate by hydrating carbon number 2? Which way is this reaction going to go, and what determines that? I'm not hiding anything from you. These are all freely reversible. What's the only thing left that can determine which direction this reaction is going to go?
If the isocitrate is used up, that's going to be favored in that
Right. In the next step, isocitrate is going to be used up. So the concentration of isocitrate is kept low, which of course pushes the equilibrium to the right. So overall, we're going to be hydrating carbon number 2 to form isocitrate, since isocitrate is going to be used up right away. Now, a conatase is a class of enzyme that we call an iron-sulfur protein, or more generally a non-heme iron protein. And we've already seen an example of a heme iron protein in the hemoglobin, right? And this, of course, had an iron. Not all iron that participates in enzymatic reactions is bound to heme, right? Sometimes they're bound to sulfur, and that's what we have here. It uses iron coordinated by inorganic sulfurs and with cysteine residues, the sulfhydryl residues on cysteines, to coordinate a bunch of irons. Okay? In this case, what the irons are doing are helping to align citrate in the active site of conatase, right? By providing coordinate covalent binding sites that can bind with the oxygens on citrate. So it's helping to position in the correct way so that the dehydration and hydration reactions can proceed. Now, other times, as we'll see when we get to the electron transport chain, iron-sulfur clusters can also be acceptors and donors of electrons. Right? We can reduce an Fe3 plus to Fe2 plus, and that Fe2 plus can then be oxidized back to Fe3 plus by donating its electrons. So these iron-sulfur clusters can help to position a substrate, or they can serve as a pathway for electrons to move through an enzyme. Okay? Now, this isocitrate is now ready to be oxidized. Okay? So isocitrate is oxidatively decarboxylated by isocitrate dehydrogenase. And the first carboxyl group leaves. It's this one in the middle. This is why I like to draw it this way, so I can keep track of which carboxyl group is going to be leaving. This isocitrate is oxidized to alpha-ketoglutarate. Notice carbon number 2 has been oxidized. We lose, of course, this as carbon dioxide. And if this is an oxidation, what else do we need? Yeah, we lost the carbon dioxide. What else do we need? If this is an oxidation of isocitrate to alpha-ketoglutarate. Right, we need an electron acceptor if we're oxidizing something. In this case, we are going to use NAD+. We reduce to NADH. And there's our reaction, catalyzed by isocitrate dehydrogenase. Right? Isocitrate plus NAD+ yields alpha-ketoglutarate plus CO2. Okay, so now we have our first decarboxylation. We've lost the first carbon. So we had 2 carbons in, now we have 1 carbon out. Luckily for us, these decarboxylations happen one after another. Okay? Succinyl-CoA is formed from alpha-ketoglutarate. Alpha-ketoglutarate is now oxidized by the alpha-ketoglutarate dehydrogenase complex, right, to form succinyl-CoA. And this is kind of strange then, right? So we lose this guy. Here we have succinyl-CoA, right? So out, we've got carbon dioxide. What else do we have coming out of here? What's the other product? NADH, right? Which means we're getting NAD plus in. Right? So we have the NAD plus in and NADH out. And we also had a coenzyme A, giving us succinyl-CoA, right? So what do we have here? This alpha-ketoglutarate is an alpha keto acid, right? Here's our alpha carbon, right? One away from our carbon number 1. Alpha carbon is a keto. And over here, we have an acyl coenzyme A, right? Where have we seen this reaction before? It's identical, with one difference. And it's on the board already. Why? And what's so similar about it? What's the structure of pyruvate? Pyruvate, right? It's an alpha keto acid. We have a keto at the alpha position. Alpha keto acid plus 2 coenzyme I, right? Yields 2CO2 to NADH. The exact same reaction. An alpha keto acid plus coenzyme A plus NAD plus yields CO2 and an NADH and an acyl coenzyme A. This alpha ketoglutarate dehydrogenase complex is almost identical to the pyruvate dehydrogenase complex, which remember is a massive complex made up of E1, E2, and E3, many copies of them, right? It's millions of kilodaltons, okay? Alpha ketoglutarate dehydrogenase looks almost identical. It's also over 1 million kilodaltons, okay? It's also regulated in the same way, and it produces the same outcome. Only difference is we started with the alpha keto acid, alpha ketoglutarate instead of pyruvate, and we end with succinyl-CoA instead of acetyl-CoA. But the process is identical, right? And we've already talked about that. Now, this succinyl-CoA has energy, doesn't it? Right? This succinyl-CoA has a thioester. So, what do we know about a thioester? It's got energy, right? It's unstable. So we can use them to generate an ATP directly. Succinyl-CoA synthetase will phosphorylate an ADP directly, giving us ATP, and now, let's see, right? And what do we have here? This is succinate, right? So this succinate, or succinyl-CoA synthetase, okay, was named for the reverse reaction, and we see this all the time. It's named for the reverse reaction because if you isolate succinyl-CoA synthetase and just provide it substrates, the delta G is such that it will go towards succinyl-CoA. It's going to hydrolyze ATP, right, form ADP and orthophosphate, and form this high-energy thioester, succinyl-CoA. Now, it so happens here where if we go backwards, we'd be hydrolyzing ATP, which releases energy, and forming this thioester, which requires energy, so it's a wash. The delta G for this reaction is actually very close to 0, okay? Which means that we can go backwards. We can hydrolyze succinyl-CoA, or not hydrolyze it, but we can form succinate from succinyl-CoA, and in the process make ATP instead of use it. Right, so this reaction is readily reversible. Again, because we are breaking a high energy here, breaking a high energy thioester, but forming a high energy compound of ATP. So far, we've made 2 NADH molecules and 1 ATP. And remember, 1 NADH molecule, the oxidation provides enough energy to synthesize 2.5 molecules of ATP. So far we've got 6 ATP made, right? 5 from the 2 NADH, 1 from succinyl-CoA synthetase, right? But now if we were to stop here, okay, we stop here, what's the problem? Why is this a cycle? Why do I have to bore you with, 3 or 4 more reactions? I don't hate myself that much, yes?
Because we have to get back to where we started.
And why do we have to get back to where we started? Couldn't we just get rid of the succinate as a waste product? Like lactate, just get rid of it? Huh? Yeah, but we've got rid of them. We lost 2 carbons already. 1 carbon dioxide here, 1 carbon dioxide there.
It require energy to do that.
Huh?
It require energy
To do what? Why? If we're building it up, now we we just have all this succinate lying around in the mitochondrial matrix, well, the concentration would be high. We don't even need an active transport. All we need is a channel to get it out, and then shoot it out into the blood and let someone else deal with it. Because what would happen after a while to the oxaloacetate? Right, we use it up. Right, we use it up, and now we have all this succinate lying around, which pushes the equilibrium of this reaction backwards, which pushes this one backwards and backwards and backwards and backwards. So eventually the entire citric acid pathway would stop, right? So the only way to keep it going functionally forever is to make everything catalytic. That's that's metabolism's favorite thing to do, is to make everything catalytic. And I know that's going to trigger some of you, and that's okay. I promise I'm not going to do a balanced reaction yet. Right? So let's get from succinate back. Succinate we can oxidize, right? So we can oxidize this carbon-carbon bond here to form fumarate, right? And we're oxidizing a carbon-carbon bond, so what should our we'll come back to that. Okay, so oxaloacetate we get from succinate. Now we're oxidizing this carbon-carbon bond here. Okay, what's our electron acceptor? Remember from chapter 15. What's the electron acceptor when we oxidize a carbon-carbon bond? FAD, right? FAD, because the reduction of FAD takes less energy. Reducing a carbon-carbon bond doesn't produce enough energy to reduce an NAD+ to NADH, but it can reduce an FAD to an FADH2. So this leaves us with fumarate, right? And this leaves us with, I'm going to draw in the hydrogens here. And now, if we look at where we are, fumarate, and where we want to get to, oxaloacetate, right? Well, what's the difference? Well, all we're trying to do, really, is to convert this methylene group, CH2, into a carbonyl, C double bond O. And we're going to see this pattern a couple of other times this semester. When we want to change a methylene into a carbonyl, this requires the same pattern of reactions: an oxidation, a hydration, and another oxidation. Sorry, right? So if we're trying to get from this CH2, sorry, too many hydrogens. I'm going to have to put them over here, CH2. When we're trying to get from this methylene on succinate to the carbonyl, all we need to do is an oxidation, a hydration, and another oxidation. So we've got our first oxidation out of the way, right? Catalyzed by succinate dehydrogenase. Succinate dehydrogenase will reduce this, or oxidize succinate. And succinate dehydrogenase is also part of the electron transport chain, right? So the FADH2 that we get here in succinate dehydrogenase is not, released out into the mitochondrial matrix and then, magically find its way to the electron transport chain. Instead, this succinate dehydrogenase is part of the electron transport chain, right? It belongs to complex 2 of the electron transport chain. So those electrons are moved directly to the electron transport chain, which again, remember, is it's going to pump those protons to generate the proton motive force, which is going to provide the energy to synthesize ATP, right? So now we've got our fumarate. Fumarase is now going to catalyze the hydration. So we've got our oxidation, now we need our hydration. Right? This is called malate, specifically L-malate. So we have our hydration there, and what fumarase does, again, it just catalyzes the stereospecific addition of this H2O. Notice it's not actually just adding an H2O, it's adding an H plus to carbon number 2 or 3, depending how you want to call it, and a hydroxyl to carbon, again, 2 or 3, depending on how you want to call it. This gives us specifically L-malate. And L-malate is the substrate specifically for the next enzyme here, malate dehydrogenase. Malate dehydrogenase catalyzes the final oxidation using an AD+. Highlighted by malate dehydrogenase. So we're oxidizing this carbon-oxygen bond to get back to oxaloacetate. And now everything cancels out, right? This citrate is made, but and it's U, so that cancels out, so is the isocitrate, and on and on and on. So if we take a little shortcut here and just say what's going in or out of the citric acid cycle, we can say that in, we've got, say, one acetyl-CoA. So acetyl-CoA plus, what else is going into the citric acid cycle? Well, we need a water over here and a water over here, don't we? Right, so two waters. What else goes in? How many? No. No. Skip right over. 3. 3 NAD plus. What else goes in? Yeah, how many? Huh? This one. All right, what else? A couple other things. H2O, got acetyl-CoA. What else? Huh? ADP, good. Let's put that phosphate there for good measure. What else? I think there's one more thing. Yeah, there's one more thing. My handwriting's not that bad. Let's go through each reaction. Acetyl-CoA, we got it. Right, H2O, we got it. Oxyl acetate, we can ignore because it's part of the cycle, so it's catalytic. Right, here we don't have anything. Here, NAD+, we got it. Here, NAD+ in coenzyme A, in. Right? You need the coenzyme A to generate the succinyl-CoA only. Right, so last thing in is CoA. And out, right, what do we get? Well, we get the 3 NADH. What else? 2 carbon dioxides plus FADH2 plus ATP. Anything else? Yeah, there's one more. Yeah, over here. A coenzyme A. So we can cancel that with the one that went in. So our overall reaction there is acetyl-CoA plus 2 waters, 3 NAD+, FAD, ADP, and alpha-phosphate give us 3 NADH, 2 CO2, right? So let's count the carbons. We got 2 on acetyl-CoA, 2 on carbon dioxide, so we're good for the carbons, and FADH2 and ATP, and that's it, right? So this is the end, non-catalytic part of the citric acid cycle, right? Which is this guy right here. Now, come back here to succinyl-CoA synthetase for just a second, right? Remember, it's irreversible, and it's this reaction here that we're talking about, the formation of succinate from succinyl-CoA. Here, there are 2 isoforms of this enzyme, succinyl-CoA synthetase. There's a liver isoform and a muscle isoform. The liver isoform actually produces GTP and uses GDP as a substrate, right? The reason for this is that GTP is used as the energy donor, right? As a phosphoryl group donor for anabolic processes. In other words, processes that build up molecules. So the liver, which sometimes has to build up fatty acids or other complex molecules, needs a source of GTP. Adipose tissue needs a source of GTP for synthesizing fatty acids or triglycerides, right? So in tissues that are performing a lot of anabolic processes, it is the liver isoform that predominates because it forms GTP. In other tissues that are performing cellular respiration, specifically, in the skeletal or heart muscle, we need a lot of ATP for energy, right? For non-anabolic processes, well, they're going to express the muscle isoform of succinyl-CoA synthetase. right? And this isoform uses ADP to form ATP, right? So let's just be careful. Now, it turns out it doesn't really matter, okay? Due to this enzyme here, nucleoside diphosphate kinase. And nucleoside diphosphate kinase simply is able to convert nucleoside diphosphates into a nucleoside triphosphate using another nucleoside triphosphate. So, if we have the liver isoform that is generating GTP, what we say is that it's functionally the same as an ATP. Why is that? Because nucleoside diphosphate kinase can take this GTP that we just made using succinyl-CoA synthetase and react it with ADP. So what are the products going to be here of this reaction catalyzed by nucleoside diphosphate kinase if this reaction is correct? What are the products here? Oh, I thought you were raising your hand.
No,
Oh. ATP and GTP, exactly, right? So if we have a GTP through the action of nucleoside diphosphate kinase, it might as well be an ATP, which is why when we talk about, for example, gluconeogenesis, and that it takes 6 high-energy phosphoryl compounds to go from 2 pyruvate molecules to glucose, the reason we don't specify, oh yeah, it's 4 ATP and 2 GTP, is because it doesn't matter, right? We can convert in between the two depending on what the cell needs. So even though there are 2 isoforms, the liver isoform, which makes GTP, and the muscle isoform, which makes ATP, functionally it doesn't matter, right? Because if the cell needs ATP, but it's got the liver isoform, they can just convert that GTP into ATP. All right, so if we look then at the end product here, the citric acid cycle just produces high energy electrons in the form of NADH, 3 of them, FADH2, 1 of them. It produces ATP, and it releases carbon dioxide as our waste product, right? So how many ATP then will we get per acetyl coenzyme A? Obviously 1, right? But what else? We have the 3 NADHs. How many ATP is that going to give us? Let's get your calculators out. What's 2.5 times 2? 5. Thank you. And what's 5 plus 2.5? 7. Hey, there we go. 7.5 plus 1.5 is what? This one's a hard one. 7.5 plus 1.5. 9. Very good. And this is the hardest one. 9 then plus the 1. 10, right? So for 1 acetyl coenzyme A, we get 10 ATP. How many acetyl coenzyme A do we get from each glucose molecule? Somebody said it. 2, right? So now we have a total of 20 ATP. Plus, how many ATP do we get from glycolysis? 2, right? And that gives us 2 pyruvate, but we just went right from glucose to acetyl-CoA. How do we get from pyruvate to acetyl-CoA? Well, we have to go through the pyruvate dehydrogenase complex. How many ATP are we going to get from there? Well, none, but we do get to what? Going from 1 pyruvate to 1 acetyl-CoA, we release a carbon dioxide, and we get also, not an ATP, an NADH, right? But we got 2 acetyl coenzyme A's from glucose, so we get 2 NADHs in the pyruvate dehydrogenase complex. So how many ATP is that? 2 NADHs? 5. Thank you. 5, right? So 20 plus 2 plus 5, right? We're not done yet. What else did we get in glycolysis? Yes, 2 ATP, but we also got, obviously, pyruvate and NADH. How many NADH per glucose? 2, right? 1 for each time we go through the glyceraldehyde 3-phosphate dehydrogenase enzyme, right? So 2 NADH times 2.5, again, is 5. So a total of 32 ATP, right? If we fully oxidize it all the way down to carbon dioxide and capture those electrons on NADH. Now, as it turns out, in most tissues, it is not 32, it's actually 30, because cytosolic NADHs kind of enter the electron transport chain a little bit weird, right? The ones from the citric acid cycle enter right away, right? They enter right away because that's where it is. It's in the matrix, right next to the inner mitochondrial membrane, which is where the electron transport chain is. So, from 1 glucose, we can get either 30 or 32 ATP, depending on which tissue we're in. All right, so moving right along here, we might actually be able to get out of here early so we can continue doom scrolling. So entry into the citric acid cycle and metabolism necessarily is controlled very tightly. Reason for this is that there are a few irreversible components that we've talked about so far. Going from pyruvate to acetyl-CoA is completely irreversible. There is no way, there is no mechanism in mammals to go from acetyl-CoA back to pyruvate, right? So once we commit those carbons to acetyl-CoA, there's only one thing for them to do, and that's to enter the citric acid cycle. So entry into the pyruvate dehydrogenase complex must be very carefully regulated, okay? Now, acetyl-coenzyme A actually also could be stored as fatty acid, which, by the way, is why eating a lot of sugar can make you fat, right? It's stored as fat. That sugar is broken down and is formed into pyruvate, fermented into lactic acid. But if fermentation doesn't occur, or we just don't need to ferment, well, then it's oxidized down to acetyl-coenzyme A, and that acetyl-coenzyme A is the building blocks for making fatty acids, right? So the sugar, those carbons are directly stored as fatty acids, right? Fatty acids also obviously can be stored as fatty acids, so it all ends up as fat, right? Which is really unfortunate. Now, the activity of the pyruvate dehydrogenase complex is tightly controlled both allosterically and via covalent modification, right? So pyruvate dehydrogenase, how is it regulated? So high concentrations of acetyl-coenzyme A and NADH, which remember are the products of the pyruvate dehydrogenase complex, inhibit the pyruvate dehydrogenase complex. And why might that be the case? Right? Let's try to think about this in terms of the citric acid cycle as a whole, right? And how this feeds back. So, if energy is not needed, what does that mean about the amount of ATP that the body has right now? Is it going to be high or low? High, right? So if the amount of ATP is high, let's come back over here, right? If the amount of ATP in the cell is high, will this process take place? No, right? Because now there's more ATP than we really need, so the concentration here kind of feeds back. If ATP then doesn't need to be made, will protons flow from here into here? No, right? Because it's going to be stopped. In order for the protons to flow, ATP must be made. But if ATP can't be made, then protons can't be made. So now this proton gradient builds up, this delta P. If the proton gradient builds up, will protons be able to move from here to here? Through the electron transport chain? No, right? Too much gradient means that not enough energy is available to move more protons and generate, an infinite gradient. So at a certain point, this stalls out. Now, if this can't pump protons, can it allow electrons to move through it? Remember, it's the electrons that are providing the energy to move those protons. Okay, so if the protons can't be pumped, the electrons aren't making their way through. Because if the electrons do make their way through, protons must be pumped. Conversely, if protons can't be pumped, the electrons can't move through. And where are those electrons coming from? NADH and FADH2, right? From here. And where are we getting these from? The citric acid. Okay. So now, if ATP levels are high, we've established that NADH and FADH2 cannot be oxidized by the electron transport chain. So now, let's come back over here to the citric acid cycle, right? In fact, so we don't have to suffer through my chicken scratch, I'll put it up on the board, all right? So we have the citric acid cycle there. The levels of NADH and FADH2 are going to be high. What is going to happen to the citric acid cycle? Slow down, I heard stop. We don't have to look past our balanced reaction that we did. This is why it's so important to be able to make a balanced reaction. ignore all that mess up there. Look at the balance reaction for the citric acid cycle. CH2A, 3NAD plus, FAD, ADP, lipophosphate yields 3NADH, FADH2 to CO2, ATP, and all this, right? What's going to happen to the citric acid cycle, which is this, if NADH levels are high? Right. So if NADH levels are high, which is a product, then feeds the whole thing back, right? We can see that in the balance reaction. We can also see that in the actual pathway. If a lot of NADH here, malate dehydrogenase cannot oxidize malate. If ADH2 levels are high, succinate cannot be oxidized to fumarate. If ADH levels are high, alpha ketoglutarate can't be oxidized. If ADH levels are high, isocitrate can't be oxidized either. Right? So everything builds back, right? Or everything slows down, goes backwards. Okay? So if everything is slowing down here, what's going to happen to all this acetyl coenzyme A? Where's it going to go? It's going to build up, right? So come back here to the regulation. Acetyl coenzyme A is building up when energy levels are high. In other words, when we don't need to be breaking down any more fuels because we already have ATP. So acetyl coenzyme A now inhibits the pyruvate dehydrogenase complex, right? By allosterically inhibiting E2, the transacetylase component, right? Remember the E1, E2, and E3. Acetyl-CoA is an allosteric inhibitor of E2 because this is signaling to pyruvate dehydrogenase that energy levels are high, oxidation of pyruvate is no longer needed, okay? But now we have all this acetyl-coenzyme A just sitting around. It can't enter the citric acid cycle. Some of it is inhibiting pyruvate dehydrogenase. Well, what's going to happen to all this excess acetyl-coenzyme A? Yeah, it's going to be stored as fatty acids. Remember, that's one of the things that we can do with acetyl-coenzyme A. So this, after a while, will then start to relieve the inhibition on pyruvate dehydrogenase once we have gotten rid of all that excess. NADH, again, we saw is going to be high when the amount of energy, the energy charge is high, right? So in this case, NADH is also a negative allosteric regulator. It's going to inhibit allosterically E3, the dihydrolipoyl dehydrogenase component of pyruvate dehydrogenase. So both of these, high acetyl-CoA and high NADH, are signaling to the cell that energy, or to pyruvate dehydrogenase, that energy generation is not needed at this time. So turn off, stop, stop using up all the pyruvate. Okay, and this is going to lead to the synthesis of glucose through gluconeogenesis due to now increased levels of pyruvate and decreased entry into the citric acid cycle. Now we can also regulate pyruvate dehydrogenase covalently, right? So we can do this actually hormonally as well. So pyruvate dehydrogenase kinase, PDK, phosphorylates E1, right? The E1 component of pyruvate dehydrogenase, the pyruvate dehydrogenase component, and inactivates it. Right? So phosphorylated E1 is inactive, and PDP, pyruvate dehydrogenase phosphatase, dephosphorylates E1, and it restores the activity. So in this case, phosphorylation is acting as a switch. Phosphorylated PDH is off, and dephosphorylated PDH is on. Both PDK and PDP are associated with that core of the pyruvate dehydrogenase complex. Remember, we showed you that big kind of spherical-looking guy, and the core was made up of E2 and E3. That's where both PDK and PDP are, right? So here we have the kinase phosphorylates active PDH, converting it into the inactive phosphorylated form, while the phosphatase hydrolyzes this off, converting it back into the active state, right? So when would we want the kinase to be active? Under what conditions might we want the kinase to be active? Just try to logic your way through it. When there's low levels of ATP, we want it to be active. Why is that? Okay, so would we want the kinase to be active or inactive in that case? ATP levels are low, we want PDH to be active, correct. So do we want the kinase to be active or inactive? What do you think? Huh? Why? We want to produce more ATP, so we want it to be over here. So we don't want the kinase to be active, right? So now we have inactive PDH, this inactive PDH. We want to instead turn it into active PDH. So we want the phosphatase to be active. Okay. So when energy levels are low, we want the kinase to be inactive and the phosphatase to be active. So conversely, if ATP levels are high, which one do we want to be active? Kinase or phosphatase? Right. And this is exactly what happens. So at rest, right, the ratios of NADH to NAD plus and acetyl-CoA and ATP are high, and these all negatively allosterically regulate the pyruvate dehydrogenase complex, as we've seen. But additionally, right, promotes the phosphorylation and inactivation of the complex by activating PDK, right? So at rest, we want the kinase to be active so we can inhibit PDH even further than we are over here, the energy charge. During activity though, high ADP activates, where are we at? High ADP here activates pyruvate kinase. Pyruvate also activates pyruvate dehydrogenase, sorry, not pyruvate kinase, pyruvate dehydrogenase, right? And calcium stimulates the phosphatase, right? Where do we get calcium from, right? Calcium stimulates the phosphatase enhancing pyruvate dehydrogenase complex. This happens during activity. Where do we get calcium from? Well, we've seen one place that we can get calcium. Huh? Right, in the sarcoplasmic reticulum. Remember, the sarcoplasm moves calcium into the sarcoplasmic reticulum, that's where it's stored. And when a signal is received from either epinephrine or glucagon on the alpha adrenergic receptor, right, that triggers phospholipase C to cleave what? PIP2, and that generates what secondary messages? Right, DHG and IP3. IP3, what does it do? Right, it opens the calcium channel. And what do we find in the cytosol of exercising muscle cells? Calcium, right? So during activity, there's plenty of calcium going around that some of it can activate the phosphatase, converting it back into the active conformation, right? Now, defects of the pyruvate dehydrogenase complex can lead to some complications, right? So, pyruvate dehydrogenase phosphatase deficiency, right, can result in a couple different conditions. But let's try to think about what's going to happen when pyruvate dehydrogenase phosphatase is deficient. What is the effect going to be on pyruvate dehydrogenase? Let's go back here, right? We have a deficiency in the phosphatase. So what's going to happen to pyruvate dehydrogenase? It's going to be mostly in the inactive state, right? Because we can't go from here to here. So what's going to happen if we have inactive PDH? Okay, we can't proceed to the citric acid cycle. Very good. At least not from glucose, right? We can still do citric acid cycle from acetyl-CoA that we get from, for example, fatty acids or amino acids, right? So what's another effect that we're going to have if we're constantly inactive with pyruvate dehydrogenase?
Just the buildup
Of?
pyruvate.
Right, we'll have a buildup of pyruvate. What's going to happen to all that pyruvate? The only other thing that can happen to pyruvate, right? Either we shuttle it out to the liver, which can convert it back into glucose, or we undergo lactic acid fermentation. And in fact, patients with pyruvate dehydrogenase phosphatase do exhibit significant lactic acid acidosis or lactic acidosis, right? The pH drops. And what happens when the pH of the entire organism drops significantly? What is that called? Anybody know? Right, ketosis or acidosis, right? And this results in neurological damage, right? This is especially detrimental to neurons. So this pyruvate dehydrogenase phosphatase deficiency actually leads to malfunctioning of the central nervous system. Right? So these tremors and all sorts of nasty side effects. Diabetic neuropathy also has similar effects, right, or reasoning, right? So diabetic neuropathy again refers to the numbness or tingling. I don't know if anybody here is diabetic. My grandfather used to get this all the time, right? Not all the time. Enough time. Anyway. So again, complication type 1, type 2 diabetes, doesn't matter what type of diabetes you have, you can get diabetic neuropathy. Now this is due to the overproduction of lactic acid by cells in the dorsal root ganglion, right? So let's think about what diabetes actually is and how this is either similar or different from PDP deficiency, right? So what is, the underlying condition kind of diabetes? Either type 1 or type 2, the effect is the same. Type 1, you can make insulin, or type 2, you're resistant to the effects of the insulin you can do. But what's the overall effect? What is insulin telling the cells to do normally? Uptake glucose, right? Okay. So, what does that mean then? Where does the glucose stay? Well, it stays in the blood. And we have less glucose in the cells. All right? So what's going to happen now in the cells? Well, there's less glucose, right? So what happens now? Nothing, right? There's not a lot of glucose to undergo glycolysis. So how is this even going to make any difference, right? Well, think about what's going to happen now. Well, now we have all this acetyl coenzyme A. All this acetyl coenzyme A that we get from, for example, fatty acid oxidation now, since we don't have enough glucose, this is going to make, or try anyway, to enter the citric acid cycle. But, what's one important thing we're going to be missing out on if we don't have enough glucose? Well, that is what an anaplerotic reaction is, right? So we'll come back to this slide in just a minute. So an anaplerotic reaction is something that refills a pathway, right? Now oxaloacetate can be used for other things, right? So oxaloacetate can be used to make glucose, which might happen in the diabetic patient, also be used to make amino acids, all right? But eventually we are going to run out of oxaloacetate because we can do other things with it, right? Here, anaplerotic, all right? So we need a way to refill oxaloacetate. And what is one way that we know already that we can make oxaloacetate? We saw it in gluconeogenesis. It's the very first step. Pyruvate carboxylase, exactly. Remember, the carboxylation of pyruvate forms oxaloacetate. Oxaloacetate then we use to make pyruvate, and then we can go all the way back up to glucose. Right? So, pyruvate carboxylase catalyzes what we call an anaplerotic reaction. Refills any oxaloacetate that might have left the mitochondria as something else, for another purpose, right? Making amino acids or making something else, right? So we need that pyruvate there in order to refill, right, in this anaplerotic reaction. And where do we get pyruvate from? Glucose. And what don't we have in our cells if we're diabetic? Glucose, right? So what's going to happen now? Well, now we still can get acetyl-CoA from other sources, but we're low in oxaloacetate because we don't have any glucose to make the pyruvate to refill the oxaloacetate in this anaplerotic reaction. So what's going to happen to all this acetyl coenzyme A? It's going to build up, right? Now what's going to happen? Well, it's going to try to inhibit the pyruvate dehydrogenase complex, as we saw before, but there's nothing to inhibit because it's not doing anything anyway. There's no pyruvate, right? So what's the only other thing that we can do with acetyl-CoA? I said a moment ago. Can be stored as fat, but even that has a limit, okay? When the concentration of acetyl-CoA is sufficiently high, and we'll see this in chapter 22, right? When this concentration of acetyl-CoA is sufficiently high, we get the formation of what is called ketone bodies, right? Have you heard of ketone bodies before, right? Well, ketone bodies are essentially condensation products between acetyl coenzyme A molecules. So this is things like acetate and acetoacetate, right? These can be used for fuels in the body, but they are ketones. They will reduce the pH of the blood. This is called ketosis or ketoacidosis. Right? This will have similar effects to what we saw in PDP deficiency. Right? It reduces the overall pH of the system and leads to neurological problems. Right? So diabetic neuropathy, because we cannot catalyze this anaplerotic reaction of making more oxaloacetate, leading to the inability of acetyl coenzyme A to enter the citric acid cycle, and instead forming these ketone bodies, which again reduces the pH of the system. Now, the citric acid cycle is also regulated at several key points. Recall that the alpha-ketoglutarate dehydrogenase complex is almost identical to the pyruvate dehydrogenase complex, so it is regulated in exactly the same way. Coenzyme A, or succinyl coenzyme A rather, and NADH and ATP all allosterically inhibit the alpha-ketoglutarate dehydrogenase complex. Isocitrate dehydrogenase is also regulated by NADH ATP levels. And this is to be expected since both isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase both catalyze the two irreversible steps in the citric acid cycle. So of course these two are going to be regulated. All right, now the citric acid cycle again is kind of this one-stop shop. Citric acid cycle integrates many of the cell's other pathways, right? So carbohydrate metabolism, fat metabolism, amino acid metabolism, and also porphyrin synthesis, which remember, heme is a part of it, right? So heme is protoporphyrin 9. So here, right, succinyl-CoA is used for the synthesis of these porphyrins, and also chlorophyll, which I'm not going to talk about because it's lame. Oxaloacetate, right, can be used to form aspartate. It's just an alpha amino acid version of the alpha ketoacid oxaloacetate. Alpha ketoglutarate is critically important in amino acid metabolism, right? So it serves as an amino group acceptor, which is the first step really in metabolizing these amino acids, right? So many citric acid cycle components are precursors for biosynthesis of key molecules. Again, specifically alpha ketoglutarate, oxaloacetate, citrate, and succinyl-CoA. All right, anaplerotic reactions. We talked about this. Fine. Now, I do want to finish off talking about the glyoxylate cycle for one important reason, in that, you know, just of general interest, if we go back here to the citric acid cycle, one thing that always kind of stands out to me is that it's actually impossible to generate a net synthesis of carbohydrates from fats, right? Which is kind of always strange to me for some reason. Remember, fats, just briefly, are broken down into acetyl coenzyme A units. So there is no equivalent of glycolysis for fatty acids, right? We break them down directly into acetyl coenzyme A. Okay? So, what does this mean? Well, now this acetyl coenzyme A from the fats is going to enter the citric acid. So, for example, palmitate has got 16 carbons. 16 or 18 carbons. Let's say it's 16, right? This is going to give us 8 acetyl coenzyme A's, right? 16 divided by 2 is 8, yeah?
Yes.
Yeah. Sorry for making fun of your math skills. So 16, so that gives us 8 acetyl coenzyme A's, right? And all these 8 acetyl coenzyme A's then are either going to be restored back as fat or enter the citric acid cycle. But we cannot generate fat, or carbohydrates rather, and that might seem confusing at first, right? So here's an acetyl coenzyme A to say we got from fatty acid. Well, we can now enter the citric acid cycle and come all the way back to oxaloacetate, which we know is part of the gluconeogenic pathway. So it seems like, well, if we can get to oxaloacetate, we should be able to get to glucose. And in fact, we can. But we cannot get to the net synthesis of carbohydrates. And why is that? So that means that the 2 carbons that enter as acetyl coenzyme A can never be stored as carbohydrate. And this is true. And why is that the case, even though we can get to this gluconeogenic intermediate here? Why can these 2 carbons never wind up on a carbohydrate? Huh?
Were they already released?
Where?
In the citric acid cycle?
As what?
As carbon dioxide.
Exactly, right? So we enter here, and we have to get all the way back to oxaloacetate, right? So we have to pass through this step, which releases 1 carbon, and this step, which releases 1 carbon. So we entered with 2 carbons, and we left 2 carbons. So So we get no net addition of carbons here, right? So we cannot store fats as carbohydrates. Now we can store carbohydrates as fat, surely, because we can get from pyruvate to acetyl-CoA, and that acetyl-CoA can be used to make fat, right? So that's no problem. And the reason again is because going here, acetyl coenzyme A, which we got from a fatty acid, we can't go backwards through the pyruvate dehydrogenase complex, right? The only place we can try to get to sugar is via oxaloacetate, but stoichiometrically we just can't do it. Plants, however, can do it. Specifically plants with oily seeds through something called the glyoxylate cycle. And they have this one critical enzyme, isocitrate lyase, that allows them to do this, right? So let's look at isocitrate at the glyoxylate cycle rather, right? So we start off exactly the same with oxaloacetate and acetyl coenzyme A, citrate synthase turns it into citrate. Conatase isomerizes this into isocitrate, so this is identical to the citric acid cycle so far. But now instead of isocitrate dehydrogenase oxidatively decarboxylating isocitrate, instead, isocitrate is acted on by isocitrate lyase. Right? What happens here is that we get a glyoxylate and a succinate. Right? This glyoxylate now can be formed into malate using malate synthase and another acetyl coenzyme A. This malate can then be oxidized to oxaloacetate. Right? So what is the net result of the glyoxylate cycle? Well, we have oxaloacetate plus H2O plus acetyl-CoA yields citrate plus OH. That's it. Now this citrate becomes isocitrate. Isocitrate becomes succinate plus glyoxylate. Glyoxylate plus another acetyl coenzyme A yields malate plus coenzyme A, malate plus NAD plus yields NADH and oxaloacetate. Did I miss anything? Probably. I'm going to assume I didn't until I'm embarrassed. Alright, oxaloacetate cancels out, malate cancels out, glyoxylate cancels out, isocitrate, citrate all cancel out. So what does this leave us with? 2 acetyl-CoA plus water yields, plus NAD plus yields 2 CoAsh, 1 succinate, what else? NADH. Yeah, that's it. Right, so let's count our carbons, make sure we didn't forget anything. Acetyl-CoA, 4 carbons, 2 times 2. Succinate, 4 carbons. Now succinate, of course, we can turn that into oxaloacetate with the remainder of the citric acid cycle. We can oxidize it back to oxaloacetate. Oxaloacetate can now go on to gluconeogenesis. Right? So here the succinate yields plus a bunch of other stuff yields oxaloacetate. So we still have those 4 carbons, and now those 4 carbons of oxaloacetate can be used to make glucose, right? Or at least half glucose. So what does this mean? That means that plants, which do express this enzyme, isocitrate lyase, can catalyze this overall reaction, which is, basically the glyoxylate cycle. 2 acetyl-CoA gives us a succinate. This acetyl-CoA comes from the oil, the fatty acid oil of oily seeds, right? So they can convert these oils into carbohydrates. And why might this be particularly important for a plant? What do plants famously need a lot of? What gives them their structure? Cellulose. And what is cellulose? It's just a bunch of glucose molecules linked beta 1,4 together. Right? So, fats, the oils of these oily seeds, are just an efficient way of storing carbons, which these seedlings can turn into cellulose and use as a structural component until they're able to carry out photosynthesis. When they can carry out photosynthesis, now they can convert carbon dioxide into sugar directly. Right? But they can't do that right away, so they need a way to get started. And that's possible here with this glyoxylate cycle, which we can't do. So we are incapable of, again, creating carbohydrates from fatty acids, again, just because we lack this one enzyme. Similar, again, to how we can't have access to all the glucose in cellulose because we lack the enzyme cellulase, right? So even the absence of one enzyme shuts off an entire pathway to that. All right. Wow, ended on time. That is remarkable. Okay, so I will see you all on Thursday. Hopefully the world's still here. And, have a good